Hi all, welcome back. We're in hearing. We're going to be talking about the sense of hearing now, or audition. And this is a much more complex sensation than either gustation or olfaction, which were in the last video. We have mechanoreceptors that are found within the cochlea in a region known as the spiral organ. And the sound waves, these mechanoreceptors, convert sound waves into electrical impulses. So this all occurs within the ear. And so let's look at the ear in a nutshell. We have this outer region of the ear known as the auricle. Then we have this external ear canal right here that leads to the tympanic membrane. The tympanic membrane is the end of the external ear. Here we have this middle ear. The middle ear contains the three auditory ossicles that vibrate when sound waves come in and hit the tympanic membrane. The malleus is attached to the tympanum, the incus is attached to the malleus and to the stapes, and the stapes is attached to the oval window. We also have this eustachian tube, which helps to decrease pressure within that middle ear, allows sound waves to move down, so electrical impulses as they move in to the cochlea, which is right here, which we'll talk about in a second. That pressure has to move back out through the round window, and then it escapes through that eustachian tube, so we don't have a buildup of pressure in this middle ear. The inner ear then contains the cochlea, which is the site for hearing. We have a vestibule, which is right in this region, and then at the tip of the vestibule, we have three semicircular canals. And so it's within the vestibule and the base of the semicircular canals where we have our sense of equilibrium, which we'll talk about in a little bit. And then you have this vestibulocochlear nerve that comes in. So you have a vestibular branch and a cochlear branch that merge together, forming that vestibulocochlear nerve. And I just talked about this. You have the external ear canal here, which has these three parts. You have the middle ear, which is right here. The middle ear is unique in that, or is special in that, it is attached to the nasal cavity and the pharynx. So you have this ear, nose, and throat that all run together, and actually your eyes then as well. So any type of infection that you have in the respiratory system can actually move into the ear canal and can cause middle ear infections as well. And so a very common disorder of children, specifically children, is otitis media, or middle ear infection. And this is because this eustachian tube in children is more horizontal, and that horizontal, the running of it horizontally and shorter than the normal tubes allows bacteria to move easier to that middle ear region, causing inflammation and pressure and pain. So typical treatment, they're going to give some type of a drop that's going to numb the ear. Sometimes they'll give antibiotics if it's a bacterial infection. And if you have multiple ear infections over a short period of time, they're going to then likely give you tubes to put in the ear. So this is a surgical procedure, laryngotomy, and what they do is they basically just put a tiny little hole in the eardrum to allow the fluid that gets in the middle ear to drain out, and it decreases the chance for ear infections in the future. If you do get tubes put in your ear, you're more prone to getting tubes put in a second time, so a lot of times you have to go in for tubes multiple times throughout your life. And as you get older, it usually is a little easier because that eustachian tube becomes more vertical and materials don't travel as effectively. The inner ear then contains the cochlea, the vestibule, and the semicircular canals. So this vestibule and the base of the semicircular canals is where the receptors for equilibrium are located. And then within the cochlea, we have the receptors for hearing. So what happens when sound waves move in from the external environment? The external ear, that auricle, is kind of like a trumpet, and it sends the waves directly into that external ear canal, and then they hit on that tympanic membrane. The tympanum is kind of like a drum that vibrates, and when it vibrates, it sends signals through the malleus, the incus, and the stapes, which then moves the vibrations into this inner region, which contains fluid. That fluid then is going to start to vibrate. And this vibration moves all the way into this cochlear region. Within the cochlea, we have a basilar membrane. So you can see this basilar membrane here. And so this is part of that spiral organ. The basilar membrane starts to flutter, and then we have these tiny little hair cells sticking out. And at the top of these hair cells, we have stereocilia. The stereocilia, so as the hair cells start to move, because this basilar membrane is fluttering, the hair cells start to move. The stereocilia is embedded in a tectoral membrane, which is like a gelatinous membrane. And so as the hair cells move, the stereocilia bend. And so here you can see it even better. There's that tectoral membrane, and there are the stereocilia. And when they bend, that converts that sound wave into an electrical impulse that is transmitted to the brain. So we're going to hear different frequencies depending on which hair cells pick up the information. So hair cells close to the base of the spiral organ pick up very high pitches, whereas low frequencies are going to be picked up at the apex of the spiral organ. And that helps us to hear different high or low pitch. So if you have damage to that cochlea, to the spiral organ, it can affect your ability to hear high or low frequencies. This next region, so the ear is the organ for hearing, but it also is your organ for equilibrium. And there are mechanoreceptors within the vestibule and at the base of the semicircular canals. There are two types of static equilibrium. Two types of equilibrium, static and dynamic. Static is the ability to maintain body position during linear movement. So think of when you're sitting in a car and you're driving forward, you don't get very dizzy just if you're sitting straight. Or moving from one level to another level in an elevator, going from first floor to second floor or third floor. That's also static equilibrium, just moving in one line. Dynamic equilibrium is then maintenance of body position during rotational movement, so when you're spinning. And so I always think of like the rides at a fair where they spin you around really fast. Or if you think about ice skaters, how they spin really fast, that would be another dynamic equilibrium. So for static equilibrium, within the vestibule, we have a region called the macula. These are specialized epithelial cells that are located in the utricle and saccule of the vestibule. These hair cells are embedded in a membrane that contains calcium crystals. This membrane is known as the otolithic membrane. And so these epithelial are embedded there. They have hair cells with stereocilia on them, just like in your sense of hearing. But in this case, they also have a long, very long hair cell, one single one, called a kinocilium. The kinocilium then is is going to, well, all the hair cells will bend, but if they bend towards the kinocilium versus bending away from the kinocilium, that will help you identify the movement that that's occurring in your body. And so as you move forward, your entire body moves forward. That's going to cause the otolithic membrane to move as well. So think of like here, we're shaking our head, we're bending our head, saying yes to someone. The otolithic membrane moves forward. This causes all of those hair cells to move and bend towards that kinocilium. When our head moves back up, the gel is going to take a little time, but it will eventually move back up as well, allowing those hair cells to straighten back out. This allows us to identify where we are, where our head is in response to the ground, and one-way movement. Whereas within the base of the semicircular canals, we have a region known as the ampulla. And within that ampulla, we have a gelatinous material, and this is at the base of each of the semicircular canals, so you can see it all throughout. There's a section called the cupula, or a gelatinous material called the cupula. Again, we have these hair cells with one lone kinocilium that's embedded within each of the hair cells. And the stereocilia are located with one kinocilium. As the body rotates, so saying no, that cupula is going to bend. It's going to start moving in one direction. As our head starts moving in the other direction, those hair cells are going to bend, and then the cupula starts to bend in the opposite direction. This allows us to recognize where our head is, even as it starts to spin. And since we have multiple canals, they all work together, allowing us to identify the direction our head is moving in at any given time. Vision is another complex process, very complex. Vision occurs in the eyes, and we have photoreceptors embedded in the innermost tunic called the retina of the eye. The types of receptors that we have rods, which are the most abundant, and they allow us to see dark vision. And then we have cones, which are less abundant, but they allow us to see bright or light vision or color vision. There's a lot of accessory structures to the eye. The first accessory we have is the eyebrow, which you can see here. And then we have eyelashes. So both of these are hairs, and hairs help to keep materials away from the eye. In this case, eyebrows keep sweat from dripping into the eye. The eyelashes will capture dust and other particles that are trying to enter the eye. And then we have eyelids. Eyelids constantly open and close in part to keep our eye moist, but also to protect our eye from any objects that are trying to get into the eye. Going a little deeper, surrounding the white of the eye on the anterior aspect, we have the conjunctiva. This is a covering that has lots of goblet cells and blood vessels. The blood vessels help to nourish the sclera, which is the avascular region of the avascular tunic. And the goblet cells help to moisten and lubricate the eye, keep the eye from becoming too dry. The cornea does not cover the iris here or the pupil, so and this is important because the blood vessels would hinder light moving in. We have lacrimal glands. I don't have a picture of lacrimal glands, so I apologize for that. But lacrimal glands are going to be found in the upper eyelid, and these lacrimal glands produce tears continuously. So we're always producing tears, and every time we blink, those tears are released and they cover the eye. They kind of wash the eye. The function of tears then lubricates, moistens, and cleans the eyes continuously. These lacrimal secretions then move into that lacrimal canal found in the lacrimal bone on the medial aspect of the eye and drain through the nasolacrimal duct to the nasal cavity. So if you've ever wondered why when you are very happy or sad or whatever and you start to cry and then your nasal canal gets all snotty, that's the reason. You have excess secretions of fluid coming into that nasal cavity causing your nose to run. Works in the opposite direction too. If ever you have a really bad cold and so materials can't leave that nasal canal because your nasal canal is stuffed up, a lot of times that the tears can't move down that nasolacrimal duct and then they fall out of the eye and it looks like you're crying because you have a cold. The eye is located in the orbit, which is composed of 6 different bones. I never ask you to identify all the different bones associated with the eye, but they're not hard to know. And then the eye is held in there. So we have the bones that protect the eye. We have a fat tissue that covers the eye. And then we have multiple muscles that help to control eye movement. So we have a superior and inferior medial and lateral rectus muscles that help to move the eye superiorly, inferiorly, medially, or laterally. Then we have an inferior oblique and a superior oblique muscle as well that helps to move the eye either up or down, so we can kind of move our eyes in a circular pattern. So moving the eye towards or away that medial aspect or down. This first outermost tunic of the eye then is known as the fibrous tunic or fibrous layer, and it has on the posterior aspect all the way up to actually the cornea is called the sclera or the white of the eye. This is composed of dense regular connective tissue. It's an avascular tissue, but it's very protective, containing lots and lots of collagen fibers. On the anterior aspect, we have a cornea. The cornea covers the iris and the pupil of the eye. The cornea has no blood vessels associated with it, so it's going to have to gain all of its nutrients from, the sclera also has no blood vessels, but it has the conjunctiva that surrounds it to feed it and nourish it. The cornea is going to gain its nutrients from the aqueous humor that's produced. The cornea functions in protection and in helping to bend light rays to the back of the eye. The next layer, known as the vascular layer, is made up of the choroid, the ciliary body, and the iris. The choroid is in the back of the eye. It's a colorful layer or a more colorful layer that takes in excess light rays. It also has lots of capillaries that help in nourishing the retina. The ciliary body is up front, and the ciliary body has both the ciliary muscles as well as ciliary process. I talked about aqueous humor in the last slide that nourishes the cornea. Aqueous humor is produced by the ciliary process. So this is continuously occurring. Fluid is being produced by the ciliary process, and that fluid then nourishes the lens, which is also avascular, the cornea, and then it drains back into the capillaries, which then process the fluid to the blood. Okay? So we're always producing new aqueous humor to nourish and then getting rid of the waste products through the cardiovascular system. Ciliary muscle then is attached to the lens through the suspensory ligaments, and these will alter the lens shape to allow us to see closer up or far away, depending. The iris is on the anterior aspect of the eye, and the iris is what most people think of as the colored portion of the eye. So when you look at someone's eyes and you're like, ooh, you have pretty green eyes, or you have nice brown eyes, the color is that iris. This is a smooth muscle, and it functions in determining the amount of light that can come in. So it controls pupil size. The pupil is just a hole. That's all a pupil is. It's not an actual thing. It's a hole that is going to change in size. So it either gets larger when there's less light out, or it gets very small when you have a lot of light coming in. And that's determined by, again, the iris. So the pupil doesn't really do anything, but the iris determines the size of the pupil and how much light will come in or go out of the eye. Then the deepest layer of the eye is the neural region, or the retina. The retina has a pigmented layer which takes in excess light rays, also produces vitamin A for our photoreceptors, rods, and cones. And then we have the neural layer, which houses those photoreceptors, rods, and cones, and takes in light rays so that we can then convert them to electrical impulses, which then our occipital lobe will change so that, or will take in so that we can perceive some type of vision. And so this is just a figure from your textbook that shows all the different parts of the eye that I was talking about. I didn't talk about the optic nerve here. So this is the optic nerve that leaves the back of the eye. Where the optic nerve leaves the back of the eye, there is a spot called the optic disc. The optic disc is also known as your blind spot. So this is where you have no rods or cones. Then over here we have a tiny region called the fovea centralis. This is the area for the best visual acuity. So this contains almost, or the majority of your cones. It doesn't have rods, but it has a lot of cones, and so when light comes in and focuses on this fovea centralis, you're going to have a perfect image there. All right, so there are 3 layers to the retina. We have the rods and cones that take in the light information. They are going to then send information, and they send information to from multiple rods or cones to a bipolar neuron. These bipolar neurons then converge together and eventually send information to ganglionic cells that form that optic nerve. I already talked about this, but I'll mention it again. The optic disc is the area where there are no photoreceptors, so this is also known as your blind spot. And this is where the ganglionic cells exit, forming that optic nerve. The macula lutea is a region just lateral to the blind spot, right here, and it contains the fovea centralis. So this is where the majority of your your cones are located and the best visual acuity. So a disorder associated with the retina, one of the more common disorders that can occur, especially with age, is a detached retina. So this occurs when the retina itself, which I don't think I mentioned, so I'm going to go back to the slide before. The retina is only held to the back of the eye at one spot, that optic disc. That's the only place that is actually attached. And we have a fluid, a thick gelatinous fluid in here called vitreous humor that holds that retina in place and helps to shape that eye. So if damage occurs to the head and causes the vitreous humor to move, that can result in a tear in the retina. The retina can actually move from the back of the eye. Since it's only attached in one spot, any movement of the retina to leave the back of the eye will result in a detached retina and in what we call floaters and this kind of visual curtain. So this can be treated through pneumatic retinopexy. This is a video that shows how pneumatic retinopexy occurs. It's very interesting. I'll let you guys watch that on your own. Another very common disorder associated with the eyesight is macular degeneration. This is probably the most common type of blindness that occurs. It can lead to blindness in developed countries. So it can occur because of high blood pressure, diabetes, which these two are often linked, or severe infections. Macular degeneration is damage or degeneration of the macula lutea, and so that area of central visual acuity is going to be gone. And so here you see that same picture with someone who has macular degeneration, where that middle image is going to be gone. So I've shown you this picture so that before, to show you how the information is taken in to the optic nerve. Now I want to look at the rods and the cones a little bit more closely. So we have a lot more rods than we do cones. Rods contain a chemical, a light pigment called rhodopsin, while cones contain different types of photoopsins. All of these need vitamin A to be produced. So retinol is a type of vitamin A, or a form of vitamin A, that is needed in the production of both rhodopsin and photoopsins, which I just mentioned. So without the retinol, without vitamin A, we wouldn't have these two pigment molecules that can take in light information, and that can lead to blindness as well. In some countries where they don't get vitamin A, a lot of individuals tend to go blind just because they can't produce the proper opsins. So here you can see the different types of wavelengths that can be taken in depending on if you're a rod or if you're one of the three types of cones. So rods take in information at about 498 nanometers of light, whereas the cones can take information in anywhere from 420 to 564 nanometers of light. And then all of these different wavelengths can allow us to see the multiple colors. So now what we're going to do, we're going to kind of change direction and go into integration of the sensory information. So I'm going to stop here so that I can start the next lecture on central processing or integration, okay?